Welcome back, everybody, for another deep dive. This time, we're going to be going microscopic. We're going to be looking at cytokine receptor families and their roles in the immune system. It's pretty amazing how these tiny proteins can actually act as messengers in our cells, orchestrating everything from inflammation to, battling those nasty infections.
Yeah. What's really fascinating to me is how they actually bridge the gap between these external signals like cytokines and then the internal workings of our cells, ultimately shaping how our immune system is going to respond to these signals.
I was reading that there are actually three main families of these cytokine receptors, type I, type 2, and then the TNF receptor families.
Right. And each of these families has their own unique characteristics, structures, and signaling pathways. You can almost think about it like they're different teams of specialists, each one equipped to handle, a particular type of immune challenge.
So let's meet these teams then. First up, we got type I and type 2 receptors. So what do they have in common and what makes them different?
Structurally, they actually share a pretty similar architecture, and both types often consist of multiple subunits, like little building blocks.
Oh, so like those modular building sets where you can make all kinds of different structures by combining those different pieces.
Exactly. And that's what allows for a remarkable diversity in these receptors that they form. However, there are some subtle but super important differences in these structural motifs that they have. For example, type I receptors typically feature a cysteine-rich domain, which is a very characteristic feature of them, whereas type 2 receptors, they have what's called fibronectin type 3-like domains. Think of these motifs like their team jerseys, signifying their unique roles.
So then how do these structural variations affect their function? I mean, do they interact with different cytokines based on their structure?
You got it. Each family has a specific set of cytokines that they bind to, almost like a lock and key. And when they bind, it triggers very distinct signaling pathways. Ultimately, that's what leads to all these different immune responses that we see. It's almost as if each family speaks a different language, communicating with a very select group of messenger molecules.
Wow. So it's not just a one-size-fits-all system, huh? There's a lot of specificity in how these receptors recognize and respond to all these different cytokines. But what about that TNF receptor family? Where do they fit into all of this?
Yeah, so the TNF receptor family, they kind of stand out a little bit because they have their own unique structural features as well as signaling mechanisms. Often they'll have those cysteine-rich domains like the type I receptors, but they also have a very distinctive death domain.
Death domain, what is that?
Yeah, so that plays a crucial role in apoptosis or programmed cell death.
Oh, wow. So they're kind of involved in deciding which cells live and which cells die. That's a pretty big responsibility.
You said it. They have a critical role in keeping that healthy balance within the immune system. Think of them as the quality control team, making sure that any old or damaged cells are removed while the healthy cells are protected.
So they're like the cleanup crew.
Exactly, making sure everything runs smoothly.
Okay, I get that. But we've been talking about these families in kind of general terms. Can we like zoom in a little bit and see how they actually work in orchestrating these immune responses?
Of course. So let's look at T cell activation, for example. Imagine a pathogen, like a virus, has entered your body. The first responders are going to be antigen-presenting cells, like your macrophages and dendritic cells, and they engulf that pathogen, break it down, and then display fragments of it, called antigens, on their surface.
Oh, so they're like the detectives. They gather the evidence and present it, right?
Exactly. And then these antigen-presenting cells, they travel to the lymph nodes. And that's where they run into the T cells, which are like the warriors of the immune system.
The T cells, yeah.
Yeah.
They're kind of like the special forces, right?
Exactly. They have specialized receptors on their surface called T cell receptors, or TCRs. They're designed to recognize those specific antigens. It's like a lock and key. Each TCR is out there searching for its match.
So when a TCR finds its match, what happens then?
When that happens, it actually triggers a whole cascade of signaling events. This includes the activation of those cytokine receptors that we've been talking about. And those receptors, they act like communication channels. They're relaying the message that there's a threat, red alert.
So it's like a chain reaction, with each step amplifying the original signal, leading to a big immune response.
Exactly. And one of the key cytokines in all of this is interleukin-2, or IL-2. When a T cell is activated, it starts making this IL-2, which then binds to receptors on the T cell itself and other nearby immune cells.
So it's like sending out a call to arms.
You got it. This IL-2 signaling, it promotes the T cells to proliferate and differentiate, so you basically get an army of specialized T cells all ready to combat that invader.
Wow. It's incredible how these cytokine receptors can orchestrate such a complex and coordinated immune response. It's like a perfectly choreographed dance, right? But I'm guessing things can go wrong in this dance sometimes. What happens if the signaling gets messed up?
That's a really important point to bring up, because when that signaling is dysregulated, it can lead to a whole bunch of health problems. Autoimmune diseases, cancer, you name it. Too much or too little activity can really throw the immune system off balance.
So it's like the music suddenly changes and everybody's stepping on each other's toes.
Perfect analogy. And one of the key pathways involved in this cytokine receptor signaling is the JAK-STAT pathway. Think of it as the superhighway of signal transduction. It takes the messages from the cell surface to the nucleus, where ultimately it controls gene expression.
So like a communication network. Cytokine receptors are the antennas, and the JAK-STAT pathway is the central hub processing everything.
You got it. And understanding how this pathway works is absolutely essential for developing therapies that can target specific receptors or even components of the JAK-STAT pathway itself.
Oh, wow. So we're not just watching this cellular dance, we're actually learning to influence it.
Exactly. We want to correct those missteps and restore harmony to the immune system.
I love that. This is so fascinating. We went from just a simple concept of families to a deeper understanding of these immune responses and the potential for manipulating these pathways for, therapeutic benefits.
It really is remarkable how these tiny proteins play such crucial roles in keeping us healthy. And it's exciting to see how our knowledge of these systems is leading to new and innovative treatments. Before we dive into the clinical applications, there's one more really cool concept that I'd love to explore.
Okay, what's that?
The idea of microdomains.
Microdomains. Like tiny compartments within our cells. That sounds like a whole other level.
It is. But it's totally worth exploring, trust me. These microdomains are thought to be specialized signaling hubs where very specific sets of molecules gather to do their jobs.
So it's like discovering hidden rooms within a familiar house, each with its own purpose.
You got it. And this concept of microdomains, it challenges how we traditionally viewed cell signaling, as just a simple linear process. Instead, it suggests a much more organized and compartmentalized system than we previously thought.
Okay, I'm definitely intrigued. I want to hear more about these hidden rooms.
Let's start with a key player in creating these microdomains, a kinase anchoring proteins, or AKAPs.
Okay. AKAPs, what are those?
Think of AKPs as the architects of these microdomains. They're designing and building these specialized signaling hubs. They do this by anchoring protein kinase A, which is a crucial enzyme in many signaling pathways, to specific locations within the cell.
Oh, so they're like the organizers of a big molecular party, making sure that all the molecules in the right place at the right time.
I love that. It's this precise organization that allows different pathways to work efficiently and specifically, even in that super crowded environment of the cell. But the question is, how do these AK keys do this? How do they anchor protein kinase A so precisely? What's their secret weapon?
Okay, you got me. What is it?
Their secret weapon is their amphipathic helix. It's this helix with two distinct personalities. One side hates water, the other side loves it.
So wait, so is this dual nature that lets them bind to certain spots and anchor protein kinase A in place?
Exactly, and there's more to it. AKPs have these specific motifs that let them bind to certain regions within the cell. Like they've got a helix-turn-helix motif that helps them bind to the cell membrane, and then they have this ACAP binding motif that lets them interact with the protein kinase A. It's like a toolkit for building these microdomains.
This is mind-blowing. It's just amazing how these tiny proteins with these intricate structures and interactions can create such a complex network within our cells.
It truly is amazing. And this whole concept of microdomains, it's changing how we think about how cells process information and respond to their environment.
I can't wait to hear more.
We'll definitely dive deeper into that in our next part. Welcome back. So yeah, I'm super excited to dive even deeper into this whole world of microdomains. Like we were saying, it really challenges that traditional view of cell signaling as just a straight line, step-by-step process. It's suggesting that it's actually this super organized system of these localized signaling hubs within the cell.
Yeah, like discovering a hidden city within our cells almost, with different neighborhoods specializing in different functions. You mentioned that there's some evidence that supports the existence of these microdomains, right? What kind of evidence are we talking about here?
Well, one of the most compelling pieces of evidence comes from all these advanced imaging techniques that researchers have now. They can actually visualize what's happening with these signaling molecules within living cells. And the images, they show these distinct clusters of these molecules, which suggests that they're not just floating around randomly.
Mm.
They seem to be organized into these specialized little compartments.
So we're actually seeing these microdomains in action.
Yeah.
Wow. Can you give me an example of what these images show us?
Yeah, for sure. Let's look at cyclic AMP signaling. It plays a role in a whole bunch of cellular processes, everything from metabolism to cell growth. Researchers have used these fluorescent probes to actually track the movement of cyclic AMP within these cells. And what they found is that it doesn't just spread evenly throughout the cell, it actually forms these distinct gradients and these localized pools, which again really suggests the presence of these microdomains.
So it's kind of like instead of having a single central control room, the cell has multiple like localized control centers, each responsible for a specific set of tasks.
Yeah, that's a really good way to think about it. And these localized control centers or microdomains, they allow the cell to fine-tune their responses to all these different signals they're getting. So, for example, a signal that activates cyclic AMP in one microdomain might trigger a totally different response than a signal that activates cyclic AMP in another microdomain.
Huh. That's really interesting. It's like having different departments within a company, each with their own specialized team and responsibilities.
Exactly. And this whole compartmentalization thing, it allows the cell to be much more precise and efficient, which wouldn't be possible if everything was just kind of floating around everywhere.
Okay, I'm starting to get the bigger picture here. Can we zoom back in on those AKKPs for a second? The architects. We talked about their amphipathic helix, but are there other things about their structure that make them such good organizers?
Yeah, so AKPs, they're really diverse in their structure and function, but the one thing they all have in common is this ability to bind to both protein kinase A and then also other signaling molecules. So it's like they're acting as scaffolds, bringing together all the necessary components for a specific signaling pathway to do its thing.
Wow, ultimate multitaskers. They're juggling multiple interactions to create these like localized signaling hubs.
I like that. And it's that precise organization that allows for very efficient and specific responses to all kinds of stimuli. For example, let's say a hormone binds to a receptor at the plasma membrane. Well, that AKP-anchored protein kinase A is right there, ready to go. And it can immediately phosphorylate its downstream targets and just trigger this whole cascade of events that lead to that specific response the cell needs.
This is incredible. It's amazing how these tiny little proteins can do all that. But how do these microdomains even form in the first place? Do they just like spontaneously assemble, or is there a more directed process going on?
That's a great question. And honestly, researchers are still trying to figure that out. It's likely that multiple things are involved, but one really interesting possibility is that the cell membrane itself actually plays a role in shaping these microdomains.
Wait, the cell membrane, how does that work?
Well, the cell membrane is not just a passive barrier. It's a very dynamic and fluid structure,
Yeah.
and it has distinct regions or domains. And those domains can differ in their lipid composition, their protein content. And these differences can influence where those signaling molecules decide to hang out.
So it's like the cell membrane is providing the blueprint for these microdomains, creating designated areas where the different signaling pathways can kind of set up shop.
Exactly. And there's studies that have shown that specific lipids and proteins can actually cluster together in the membrane and form these platforms for signaling complexes to form.
This is all making a lot more sense now, although it feels like we're only scratching the surface of this whole topic.
Yeah, absolutely. We still have so much to learn about how these microdomains are formed and regulated and how they actually influence everything the cell does. But one thing's for sure, understanding these little signaling hubs is crucial if we want to unravel all the mysteries of how cells communicate and how we can develop new and more targeted therapies for so many diseases.
Totally agree. It's been such an eye-opening journey so far.
It has.
Welcome back. So we've been on this incredible journey exploring cytokine receptors and their signaling networks. We've talked about those different families of receptors, how they orchestrate those immune responses, and then we went deep into this concept of microdomains, those localized signaling hubs within our cells. But let's shift gears a bit and talk about the real-world implications of all of this. How can understanding these, microscopic processes actually benefit patients?
Yeah, that's a million-dollar question, right? And it's one that researchers are working really hard to answer. One of the most exciting prospects, I think, is the potential for developing much more targeted therapies. If we can actually pinpoint the exact location and the molecules that are involved in, say, a disease process, we could design drugs that act like, those precision missiles. They would only target those specific components.
So instead of using like a sledgehammer to break down a whole wall, we could just use a scalpel to remove a single brick.
Precisely. And that's what's so cool about microdomains. They offer this potential to fine-tune our interventions. We can target specific events within a cell instead of just broadly activating or inhibiting a whole pathway.
Can you give an example of how that would work?
Sure. Let's think about autoimmune diseases. That's where the immune system is attacking the body's own tissues. And a lot of times that involves dysregulation of cytokine signaling. It leads to too much inflammation and damage to those tissues.
So like the immune system's dance has gone totally haywire and it's just wreaking havoc.
Exactly. And traditionally, the way we've treated autoimmune diseases is by just generally suppressing the immune system. But that comes with a whole bunch of side effects, right?
Yeah. It's like shutting down the whole party just to stop a couple people from getting out of control.
Right. But with a deeper understanding of these microdomains, we might be able to develop much more targeted therapies. For instance, if we could identify the specific AKPs and signaling molecules involved in, let's say, a particular autoimmune response, then we could potentially design drugs that just disrupt those interactions without messing with other immune functions.
Oh, so we could target those specific troublemakers without affecting the whole party.
That's the idea. And researchers are already looking at this approach for a bunch of conditions. They're developing drugs that target specific AKPs involved in inflammatory responses, hopefully reducing inflammation without compromising the immune system's ability to do its job.
That's amazing. Are there other areas where this could be helpful?
Oh, for sure. Cancer treatment is another really promising area. Cancer cells, they often hijack those normal signaling pathways, including those involving cytokine receptors and that JAK-STAT pathway, to basically grow out of control and survive.
So they're like rewriting the choreography, turning a nice waltz into like a crazy mosh pit.
Perfect analogy. And by understanding those specific microdomains and signaling intents that are driving that cancer cell behavior, we can develop drugs that just disrupt those processes without harming the healthy cells.
It all sounds pretty incredible, but are there like challenges in actually translating this knowledge into, effective therapies?
Yeah, of course. It's definitely not easy. One of the biggest challenges is just the sheer complexity of these systems. I mean, we're talking about these intricate networks of molecules interacting with each other. Pinpointing those exact targets for intervention is a really tough job. Another challenge is actually getting those drugs to reach those microdomains within the cells. They're often tucked away in these specific compartments, and it can be tricky to get the drug to where it needs to go.
So like navigating a maze to deliver a package to a specific apartment.
Exactly. But even with all those challenges, I think the potential benefits are just too big to ignore, you know? The possibility of developing these highly targeted therapies that can effectively treat diseases with way fewer side effects, that's what keeps researchers going.
Yeah, I can totally see why there's so much excitement about this. It feels like we're on the edge of a whole new era of medicine.
I think so too, and it's all because of this deeper understanding of these fundamental principles of how cells talk to each other, from those different cytokine receptor families to this whole concept of these microdomains and their organization. It's really a testament to how powerful scientific inquiry can be. There are so many wonders waiting for us when we really look closely at what's happening inside our cells.
Well, this deep dive has been absolutely mind-blowing. I feel like we've gone on this incredible journey through the immune system, discovering all these hidden compartments within our cells, and getting a glimpse into how all of this could change how we treat diseases in the future.
It's been a pleasure talking about it with you, and I hope our listeners have enjoyed it too. I hope it's sparked a sense of wonder in everyone. There's just so much beauty and complexity in life at the molecular level.
I couldn't agree more. To all our listeners out there, keep exploring, keep asking those questions, and keep that sense of wonder alive. We'll see you next time for another deep dive.